This laboratory experiment implement molecular identification technique utilizing mitochondrial cytochrome oxidase subunit 1 (COI) gene as a DNA barcoding marker. The COI gene is one of the most popular markers used for molecular systematics, with fragment near its end frequently utilized to inter-phylogenetic relationship in alignment with the standard of the DNA barcoding consortium. To analyze this marker, DNA isolation and target region amplification are performed using a polymerase chain reaction test. This method relies on enzyme-specific COI primers, a C-type buffer solution, and thermal cycling to exponentially replicate the target DNA sequence. Following amplification, gel electrophoresis are performed to separate the resulting mixture of biomacromolecules, which can include DNA, RNA, and protein. Finally, the agarose gel is viewed under a UV transimulator to observe and verify the successful formation of the DNA band.
For materials and methods for DNA isolation protocol, CTAB method, the casting tray was placed inside the gel. Electrophoresis tank filled with TAE buffer. Load the samples. The samples were loaded into the wells of the gel. Electrophoresis was run at 95 V and 200 mA for 35 minutes. The agarose gel was soaked in ethidium bromide solution for 1 minute. The gel was viewed under a UV transilluminator to observe the DNA bands. gel preparation and polymerase chain reaction, PCR. A 25-ol master mix was made using sterile water, TAC buffer MgCl2, DNTPs, COI primers, TAC polymerase, and the DNA template. The machine was heated to 92 degrees Celsius for 5 minutes to separate the DNA strands. The machine repeated 3 steps for 35 rounds. For denaturation, 92 degrees Celsius for 1 minute. For annealing, 55 degrees Celsius for 1 minute, primers bind. And for extension, 72 degrees Celsius for 1 minute, DNA copies. Final extension, the machine did a final copy step at 72 degrees Celsius for 5 minutes.
Results and interpretations. DNA samples are placed in gel walls and run using an electric current. The samples go into the gel holes and electricity is turned on to start the separation. The goal is to sort and view DNA pieces based on their size. This method acts as a visual tool to separate and identify different DNA fragments lengths. First, DNA is extracted from the sample and target gene is copied. Genetic material is isolated from the material and the mitochondrial COI gene is multiplied using PCR. Next, the copied DNA moves through the gel, sorting by size. The PCR products travel through the gel pores. Smaller DNA pieces move much faster than larger ones. A chemical stain is used to make the DNA bands visible. Adding a special dye allows final positions of the sorted DNA to light up clearly under UV light. The DNA profiles can be checked against known standards. Researchers can easily match the sample's band pattern with verified reference markers. The unique genetic fingerprint can be compared to official database. This allows researchers to cross-check the DNA result with online digital libraries to find an exact match. Using this DNA sequencing makes identification highly reliable. This method gives researchers an accurate identification even when the sample is damaged, immature, or impossible to identify by eye. Finding the exact match helps protect crops. Correctly identifying the sample allows researchers to develop fast and effective management plans. The first interpretation is bands presence. Distinct bands are visible, showing that genomic DNA was successfully isolated. The lens on the far left lacks these clear top bands. The second is band intensity. The visible bands are highly intense and bright, which indicates a high concentration of extracted DNA. The rightmost lens shows a complete loss of intensity or very faint trails. Unclear bands. Heavy smearing is visible below the main bands. This means the DNA was likely degraded or overexposed under UV light.
For our PCR result, the result did not show any target DNA bands for the samples on the agarose gel. A DNA ladder is visible on the bottom left, but no amplified fragments appeared in the sample lanes. This suggests that the PCR amplification failed, possibly due to poor template quality or incorrect annealing conditions. The main difficulty for us while conducting this experiment was the difficulty in loading the samples due to our shaky hands, which also made it challenging to precisely pipette the samples into the tiny gel wells. This risks the cross contamination and loading into the wrong well. Next, we also face difficulties in operating inside the laminar flow hood, where because it made us feel awkward and difficult to maintain a strict sterility to prevent contamination.